So today I want to talk to you about the growth rates of Lampsilis cardium, an old favorite of all of ours, in terms of the differences between males and females and as a function of different environments that they live in. My name is Mariah Scott, and I'm a PhD student at the University of Chicago, and my advisor is Rüdiger of the Field Museum. So I think we all have our own opinions about babies, but we can probably all agree that they are very expensive. And freshwater mussels, the females can only give so many resources to their offspring before they're really going to start hindering their own growth and even survivability. I'm really interested in looking at the growth rates of males and females, same species, same environment, to get an idea of how expensive is reproduction for females in comparison to their male counterparts. So I'll be looking for significant growth rate differences between females and males of these species. We do know in terms of the shell shape, the females are sexually dimorphic from males. They tend to have shorter length and a puffier posterior end. And I'm going to be looking also at a couple habitats to be seeing if this amount of differentiation holds between different rivers, specifically the Embarrass River and Sangamon River of Illinois. So I have some museum specimens. I have some field collected shells from a few rivers. So I start by doing a 3D scan of the shell using an optical scanner, and then also Archimedes' buoyancy principle to get the shell volume. I then section the shell to identify annuli. Now these are annual growth rings that are laid down in temperate environments during the winter when the shell growth slows. So you can see in cross-section 2C, there's 5 annuli. This individual has experienced 5 winters. I then combine the 2D and 3D data to get a sense of where these lines are on the shell, and then can make measurements at successive annuli, so essentially yearly growth of the individual. With this, I can get the shell length, external surface area, and volume of the whole shell with individuals, and then have that measured as a function of how many annuli they have. I can also look at successive changes in the length of the shell and external surface area of individuals as a function of their yearly ontogenetic or over the course of their life growth. I can make these comparisons between males and females of the same environment, and also kind of look between environments of if we're seeing differences there. So when I'm looking at whole shell measurements, all 5 came back with significantly different growth rates in terms of von Bertalanffy growth models of males and females. And this was all the types of measurements that I did. Now we can see when you look at the ontogenetic growth in figure 6, that yeah, we're seeing the same result of males are bigger than females, but in these preliminary results, we're actually seeing that the divergence kind of happens after annuli 3. So I'm very excited to continue the study, get more results, and would like to thank my funding sources as well as Ashley Vance for helping identify the annuli.